going to turn off, so I had to find the charger. Anyways, after explaining the renal cortex in the end of the last video, now we're going to discuss the medulla. It's defined as the region that lacks the renal corpuscles. So the region of the kidney that does not have these corpuscles right here is known as the renal medulla. It's the dark inner portion of the kidneys. It contains renal pyramids, which are conical bodies. These renal pyramids are basically these cone-shaped structures right here. So these are renal pyramids. They're conical bodies, as you can see, and these are found in the renal medulla. It's the dark inner portion, as we said, and it contains most of the length of the loops of Henle and most of the collecting ducts. Remember, we said that the renal cortex is defined as the region that contains the corpuscles. In contrast, the renal medulla contains most of the length of the loop of Henle, so this part right here is found in the medulla, and most of the collecting ducts. These are also found in the medulla. Other than that, that's all we need to know about the medulla for now. We need to know about the renal papilla. If you remember, when we talked about the heart, we said the heart was unique in that its bottom part was known as the apex, and the top part, the one that connected to the great blood vessels, was known as its base. Here we have a similar concept. The renal papilla here, which is the point of connection between the renal pelvis and the pyramids, is known as the apex of a renal pyramid. So the papilla here is the apex of the renal pyramid. It connects to the calyx. The calyx are basically branches of the renal pelvis. So first of all, let's discuss the renal pelvis. The renal pelvis joins the concave part, النقطة المقعرة, of the kidney with the ureter, which drains to the bladder. So these are the ureters. So the renal pelvis joins the concave part of the kidney with the ureter. It branches out in right angles or in an oblique manner. Oblique means neither right angles or perpendicular. As it moves away from the cortex, towards the cortex, as it branches out, we can classify it into major and minor calyx. The major calyx, right here, are the parts which branch out directly from the base of the renal pelvis. The minor calyx are the second branching out, which branches away from the renal pelvis that are smaller in diameter. So basically these parts here are the major calyxes branching out from the renal pelvis directly, but the farther away the branches get, they become minor calyx, which are smaller in diameter. So this is basically the calyx, which is connected to the renal pyramid via the papilla, which is found at the apex of the renal pyramid. Then we have the renal pyramids, which we said are found in the medulla, which are conical bodies found in the cortex. This is an incorrect part. It's actually found in the medulla, so please fix this. It's not found in the cortex. It's found in the medulla. The renal columns are the spaces between the renal pyramids. So the spaces here between each pyramid is known as the renal columns. Outside of the kidney, we have the fibrous capsule. It's tough and fibrous, as you can tell from its name. It's fibrous because it contains relatively large collagen fibers arranged in a two-dimensional array. It's covered by mesothelium on surfaces exposed to the peritoneal cavity. If you remember from the serosa, we said that mesothelium is basically simple squamous epithelial tissue. And I just mentioned it here because it came up twice. So the fibrous capsule is covered by mesothelium, which is simple squamous epithelial tissue. It consists of two layers. The inner layer of the capsule is made up of a fibroblast layer, or cells called fibroblasts, that separates the outer layer from the parenchyma of the cortex, and the outer layer is known as the outer layer. The manual mentions that the myofibroblast layer, or the inner layer, might be absent in smaller mammals. For example, the kidneys of mice might lack this inner myofibroblast layer. This is basically all we need to know about the kidneys. Now, when it comes to the liver, we need to know a couple of things. The most important is that it's divided into 4 lobes. I've often seen this question come up. which will be asked directly. How many lobes does the liver have? It has 4 lobes. It's the largest gland in the body. It has both exocrine function and endocrine function. By exocrine function, we mean that it releases the chemicals through ducts to another surface within the body. And by endocrine, we mean that it releases the chemicals directly into the bloodstream or tissue. How is it an exocrine gland? Because it secretes the bile into the duodenum via the ducts, via a system of bile ducts. So that's how it's exocrine. And it's endocrine because it synthesizes and releases organic compounds directly into the bloodstream. So this is why we say that the liver has both exocrine, it secretes through ducts, and endocrine, releases directly into the bloodstream function. What is the function of the bile canaliculi? The function of the bile canaliculi is to collect the bile secreted by hepatocytes. Before I continue here, let's discuss the descriptions here. First of all, this is a hepatic lobule, or a liver lobule. Now, I noticed that in the manual they also referred to liver cells as acini, but they meant a group of cells. Now, in some textbooks, they mention, first of all, the liver lobule is a functional unit of the liver. In some textbooks, instead of lobule, they say acini. So, lobule basically means acini. This whole thing is the functional unit of the liver. We have in the middle a central vein, which forms the axis of the hepatic lobule. It receives blood from the sinusoids and drains into intercalated or sublobular veins. What are the sinusoids? They, according to the manual, form an extensive fenestrated system of vascular channels. So they have blood vessels and they radiate, yushiru, from the central vein. We have to know that they're lined with endothelial cells and Kupffer phagocytic cells, and they receive blood from interlobular branches of the portal vein and the hepatic artery. The blood flows to the center of the lobule and is drained by the central vein. So this central vein collects blood from the sinusoids. Now, the next thing we're going to talk about is the space between the sinusoids. As you can see here, the sinusoids are the white parts. The space between them are the hepatic cords, which are sheets or rows of hepatocytes separated by sinusoids. They are the ones that contain the bile canaliculi. What are the bile canaliculi? They collect the bile secreted by the hepatocytes. So the bile canaliculi are found within the hepatic cords, which are made up of hepatocytes. And their function is to collect the bile secreted by the hepatocytes. What are the hepatocytes? Hepatocytes are also known as liver cells. Hepato or hepatic means to do with the liver, and cyte means cells. They are the main tissue cell of liver. Other liver cells we talked about are the endothelial cells and the Kupffer phagocytic cells that line the sinusoids. They are polyhedral with a round nucleus. They're arranged in cords and plates known as hepatic cords that radiate from the central vein in a spoke-like manner. So, similar to the sinusoids, both the sinusoids and the hepatic cords radiate from the central vein. Spoke-like, if you remember, we discussed in class, spokes means the way that a bicycle wheel looks like. So they look like basically a bicycle wheel in the way they're arranged. We need to know that the hepatocytes contain large amounts of rough endoplasmic reticulum and free ribosomes, and they're involved in protein synthesis, protein storage, and transformation of carbohydrates, as well as in synthesis of cholesterol, bile salts, and phospholipids, as well as detoxification, modification, and excretion of exogenous and endogenous substances. So that's the hepatocytes. The hepatocytes are arranged in cords and plates known as hepatic cords. These hepatic cords contain the bile canaliculi, whose function is to collect the bile secreted by the hepatocytes. In between the hepatic cord, we have the sinusoids, which carry blood towards the central vein where it's drained. In between the lobules, we have this part, which is the portal space. It's the space between the hepatic lobules. It contains a branch each of the bile duct, the hepatic artery, and the portal vein. The hepatic artery and the portal vein are important because they transfer blood to the they transfer blood to the sinusoids. And this is basically, oh, this is the final part. What is bile made of? It's made of bile salts, water, bile pigment, cholesterol, lecithin, fat, and inorganic salts. As we mentioned in class, we need to know that bile salts are the only part of the bile to be recycled. What is the function of the bile that's secreted by the hepatocytes? The main function of the bile is twofold: to accelerate the action of pancreatic and intestinal lipase, which basically means to increase the action of enzymes, and to facilitate the absorption of fats from the intestine. That's everything we need to know about the human liver. After the human liver, we have the last two organs, spinal cord and trachea. The spinal cord, the human spinal cord, is basically, along with the brain, it forms the CNS, or central nervous system. We need to know that it's divided into 31 pairs of nerves, or 60, which are 62 in total. The nerves are named according to where they leave the spine, so we have 5 groups. Now, in class, I told you that the first one is cranial. It's actually cervical. It's a typing error in the manual. The first 8 pairs are in the cervical region, not the cranial region. The cranial nerves are separate, as we saw in the final lab, the systems lab. So 8 pairs are in the cervical region, 12 pairs are in the thoracic region, 5 pairs in the lumbar region, 5 pairs in the sacral region, and 1 pair in the coccygeal region, which is the tailbone or the coccyx. Now, what does the spinal cord contain? It contains white matter, which travels in tracts or in rows to and from the brain, and it contains the myelinated axons. Gray matter is edge-shaped, and it contains neuroglia, neurons, glial cells, and non-myelinated axons. We also need to know that on either side of the spinal cord, we have 3 horns: the ventral horn, the lateral horn, and the dorsal horn. The ventral horn contains something known as somatic motor nuclei. The lateral horn contains autonomic motor nuclei, and the dorsal horn is divided into the somatic sensory portion and the visceral sensory portion. That's all we need to know about the human spinal cord. Finally, we have the human trachea. The human trachea is also known as the windpipe. It's 4 inches long and less than an inch in diameter for most people. It begins just under the larynx. The other name for larynx is the voice box. It runs down behind the breastbone, which is the sternum. It divides into 1 bronchus for each lung. There are 16 to 20 cartilage rings that line the trachea, with narrow membranous regions between each ring. The back part of each ring is made up of muscles and connective tissue. Let me show you the diagram so it becomes clearer. Here we have the walls of the trachea, the tracheal wall. As we can see here, the back part of the ring is made up of muscle and connective tissue. So this back part here is made up of muscle and connective tissue. First of all, just to make it clear, this ring right here is the esophagus. So just to clarify what this is the esophagus, and this is the trachea. This part is the trachea. So here we have the trachea, and we can see that there are 4 walls of the trachea. So first of all, before we move on to the walls, the back part of each ring is made up of muscles and connective tissue. And we need to know that the trachea widens and lengthens slightly with each breath in, and returns to normal or resting size with each breath out. So what is the tracheal wall made of? It is divided into 4 layers. The innermost layer is the mucosa, and the outermost layer in this case is called the adventitia. First of all, the mucosa, as we can see here, the cells here are ciliated, and they're separated by goblet cells. The cells here are pseudostratified because they're supposedly not all found at the base. The nuclei are not all ovoid in shape, and they're not all arranged at the base. So the mucosa consists of ciliated pseudostratified columnar cells with many goblet cells in between. The function of the goblet cells is to produce sticky mucus to coat the inner lining of the trachea to catch any debris present in the inhaled air. The cilia found on the surface of the pseudostratified cell functions to beat together to push the mucus away from the lungs. What happens to this mucus? One of 2 fates. The mucus, along with the trapped contaminants, is either expelled through the larynx by coughing, or swallowed and digested in the stomach. So that's what happens to the mucus. that comes out of the tracheal wall. The next layer is the submucosa, which is this part right here. This second layer is the submucosa. It is made up of areolar connective tissue. Another word for areolar connective tissue is loose connective tissue. It contains blood vessels and nervous tissue that provide support to other layers of tracheal wall. Many collagen and elastin and reticular protein fibers found in the submucosa give soft support and elasticity. We also have longitudinal smooth muscle fibers in the posterior traces between the ends of the cartilage rings. And we have smooth muscle tissue, which allows the trachea to adjust its diameter as needed. When we're breathing in and out, the diameter of this ring increases and then decreases. The third layer is the hyaline cartilage. These are C-shaped rings. The whole thing looks like this. They're C-shaped rings, as you can see here. And their functions, as is the function of any cartilage, is to maintain an open airway. It's resistant to external stresses, and the parts in which chondrocytes are found are known as the lacunae. This is a piece of information in the manual. Finally, we have the adventitia, which here is a layer of areolar connective tissue that loosely, its function is to anchor the trachea to the surrounding soft tissue. Now this is adventitia here because it has a layer of areolar connective tissue, or it's adventitia because it's connected to which type of organ? If we go back here, we can see that adventitia is connected to the organs that are retroperitoneal, meaning the ones that are not bound to the abdominal wall. It is called, it does not have mesothelium. So here, the adventitia here is only made up of loose connective tissue without any mesothelium. But if we go back to the intestinal wall, the serosa here, if we're talking about the serosa, does have mesothelium. I hope this proves to be useful to you and that it helped you better understand this lab.